Good morning, friends. How is everybody doing? You guys ready to get into module number 7? I am happy to help. All right, here we go. This is module number 7. The title is Cellular Reproduction and DNA, and it begins on page 195. All right, here we go with the introduction. As we learned way back in module number 1, all living organisms reproduce. Indeed, the ability to reproduce is one of the 4 criteria for life. It makes sense then that we should study this process in great detail. As is the case with many of the concepts in biology, however, reproduction is a difficult and detailed process to study. Thus, we will spend both this and the next module exploring some of the facets of reproduction. The process of reproduction has always fascinated scientists. Throughout the history of biological inquiry, scientists have tried to understand how an organism's offspring receive the traits and characteristics that make them what they are. Sometimes, an organism's offspring might look identical or nearly identical to its parent or parents. In other cases, the offspring and parents look so different that you can hardly believe the offspring came from those parents. When we look at a child, for example, we usually try to find the characteristics that the parents and child have in common. When we see that a boy and his father both have thick brown hair, we say that the boy has his father's hair. When we see that he and his mother share vividly green eyes, we say that the boy has his mother's eyes. Sometimes, however, we have a hard time finding such similar traits. For example, 2 brown-haired, brown-eyed parents can have a child with blonde hair and blue eyes. How does that happen? Well, the explanation for these facts eluded scientists for centuries. To be sure, there have always been theories that attempted to explain how characteristics pass from parent to offspring, but many were quite ridiculous. For example, there were those that thought characteristics like the height, strength, and weight of an offspring were determined by the activities of the parents. These scientists, called Lamarckian biologists, thought that if parents spent a lot of time in strenuous activity, their children would be born stronger. Somehow they thought that the parents' activities would send messages to their reproductive systems, telling them what kind of offspring to produce. The beginning of a real explanation for how traits are passed on from generation to generation was developed by a monk named Gregor Mendel in the mid-1800s. He did careful experiments with peas, determining how pea plants reproduced and passed on characteristics to their offspring. Unfortunately, this magnificent work was tucked away in a monastery for more than 50 years. When it was finally discovered, it laid the groundwork for all of our modern studies of genetics. Genetics, the science that studies how characteristics get passed from parent to offspring. Genes, chromosomes, and DNA. Building on the work of Gregor Mendel, scientists today have determined that each organism contains a storehouse of information that governs its traits and characteristics. As we have already mentioned, this storehouse is called DNA, and its main function is to tell the cell what proteins to make. Although it sounds hard to believe, most of your traits and characteristics are governed by what kinds of proteins your cells make. Your eye color, for example, is completely dependent upon what proteins are produced in some of the cells in your eyes. Thus, by coding for the production of certain proteins in your eyes, your DNA determines your eye color. Now, of course, not all of your traits and characteristics are completely determined by your genetic makeup. For example, if you lift weights and work out, you will develop strong muscles. You might think then that your muscle strength is not determined by your DNA. Well, this is really only partially correct. Even though you can increase your strength by working out, there's a fundamental limit to how strong you can become. For example, a person might lift weights and work out every day, but never become as strong as another individual who really does not exercise seriously at all. In the case of muscle strength, your DNA determines a general range of how strong your muscles can become. If you do not exercise your muscles, you will stay on the weak side of the muscle strength range determined by your DNA. If, on the other hand, you work out seriously, you can increase your muscle strength to the strong side of the range your DNA has determined. You might never become as strong as you want to become, however, because there is some inherent limit set up by your DNA. It turns out that the majority of characteristics in your body are determined in this way. The DNA sets a range of possible characteristics, sometimes called your genetic tendency. And your activities determine what portion of that range actually manifests itself in your body. Some people have a genetic tendency to be overweight. If they eat just a little more than they need, they will immediately gain weight. Others, however, have a genetic tendency to stay slim. They can eat almost as much as they like and never gain a pound. Although such tendencies exist, they can nevertheless be controlled. Even if you have a genetic tendency to be overweight, you can keep your weight down with strict diet and exercise. Thus, your DNA sets a general tendency, but you can work to fight that tendency if you wish. You might have read or heard that scientists have discovered that alcoholism is genetic. This is, in fact, partially true. A large study in Sweden, for example, looked at alcohol use in 3,000 children who were adopted and raised by non-relatives. The incidence of alcoholism was 2.5 times higher among those children who had at least one biological parent who was an alcoholic. What does this tell us? Well, consider an alcoholic whose father was also an alcoholic. You could argue that he inherited his alcoholism from his father through his DNA. Or you could argue that he became an alcoholic because his father's actions trained him to be an alcoholic. In this study, however, the parents who had the children did not raise them. Thus, if alcoholism wasn't at least partly genetic, you would expect the rate of alcoholism to be independent of the child's biological parents. It wasn't. That tells us alcoholism has a genetic component. However, there are plenty of people whose parents are alcoholic who are not alcoholics themselves. And there are plenty of people who are alcoholics but have no family history of alcoholism. Thus, the data seem to indicate that DNA might set up a tendency toward or away from alcoholism, but that people can break that tendency. The main point to this discussion, then, is that your DNA alone does not determine who you are or what you will become. There are some traits, blood type, for example, that are completely determined by your DNA. For many of your characteristics, however, your DNA just sets up a general framework. Within that general framework, what you do with yourself will ultimately decide what you become. Since DNA is not the only factor in determining what kind of person you will be, we generally say that there are three factors in determining the characteristics of a person: genetic factors, environmental factors, and spiritual factors. Genetic factors: the general guideline of traits determined by a person's DNA. Environmental factors: those non-biological factors that are involved in a person's surroundings, such as the nature of the person's parents, the person's friends, and the person's behavioral choices. Spiritual factors: the factors in a person's life that are determined by the quality of his or her relationship with God. These three factors each work together to determine what kind of person you are. There's no real consensus in the scientific community as to which of these factors is the most important, if any. Needless to say, however, from the Christian point of view, spiritual factors are of the utmost importance. Since spiritual factors do not really apply to any organism other than humans, we generally say that the characteristics of animals and plants are determined by genetic and environmental factors. Even though we do not know much about the relative importance of genetic factors and environmental factors, we can make one statement for sure: the genetic factors are laid down first. From a scientific point of view, these are the factors most easily studied, so we will concentrate on them in this course. The information in an organism's DNA is split up into little groups known as genes. Although a firm definition for a gene is rather hard to pin down, the following will work for the purposes of this course. Gene: a section of DNA that codes for the production of a protein or a portion of protein, thereby causing a trait. As we mentioned before, your genetic traits or tendencies are determined by what proteins are produced in your cells. Thus, by coding for the production of a certain protein, a gene is actually coding for a particular genetic trait or tendency. How does a gene code for the production of a protein? You will learn about that in the next section. For right now, we want you to have a bit of experience with DNA by performing the following experiment. Now you know what the strands of DNA look like on a macroscopic scale. If all of the DNA from a single human cell were extracted in this way, you would have about 2 yards of strands. Protein synthesis, part 1, transcription. You might be wondering how instructing cells to make proteins can result in the traits that DNA gives us. If you're wondering that, perhaps you don't understand how important proteins are. Remember, in multicellular organisms, specific cells have specific tasks which they must accomplish so that the organism will survive. In your body, for example, you have skin cells that shield the inside of your body from contaminants. You also have cells in the retina of your eye that detect light and send electronic messages to your brain based on the light that they detect. How in the world do these cells perform such radically different tasks? The answer is simple. They produce different types of proteins. By and large, the tasks that a cell can complete are dependent on the proteins that it produces. If it produces one set of proteins, a cell functions as a skin cell. If it produces another set of proteins, it acts as a cell in the retina. So, how does a cell know what proteins to produce? That's the function of a gene, and that's what you will learn in this section. Before we can show you how a cell uses DNA to produce proteins, we first need to give you a little more background on an important chemical of life, ribonucleic acid. We briefly mentioned this molecule in the previous module, but now we need to spend a little more time on it. As you might have already guessed by virtue of its name, RNA must have some relationship to DNA. Well, it does. In fact, there are a lot of similarities between them. RNA is also made up of nucleotides, but the individual structure of the nucleotides is a bit different. First of all, the sugar that makes up the foundation of the nucleotides is ribose, not deoxyribose, as is the case with DNA. Like DNA, the nucleotides of RNA join together in long strands. Unlike DNA, however, they do not form a double helix. Instead, RNA is usually a single strand of joined nucleotides. Like DNA, RNA has 4 nucleotide bases. While 3 of them are the same as DNA's, adenine, cytosine, and guanine, one of them is different. Uracil. Uracil performs the same tasks in RNA that thymine does in DNA. Thus, RNA has uracil in place of thymine. This can be confusing to students, but don't let it stump you. When you're thinking about RNA, just remember that uracil takes the place of thymine. It turns out that DNA is much more chemically stable than RNA because DNA uses thymine and deoxyribose instead of uracil and ribose. This means that it is unlikely to undergo major changes with time, making it the ideal molecule for storing information from generation to generation so that each organism reproduces after its kind. Genesis 1:11. So, RNA has a lot of similarities to DNA, but also some differences. Why is it important in the process of how the cell makes proteins? Well, one kind of RNA acts a lot like a camera. You see, the main portion of a cell's DNA is in its nucleus, but proteins are made in the ribosomes, which are outside the nucleus. In order to get information from the nuclear DNA to the ribosomes, one type of RNA makes a snapshot of the DNA and takes that information out of the nucleus to the ribosome. How does the RNA do this? Well, first you need to remember something from module number 5. When we told you about DNA in that module, we told you that certain nucleotide bases can only link to other nucleotide bases. For example, cytosine can link only to guanine, and thymine can link only to adenine. Well, RNA has nucleotide bases as well. It has cytosine, guanine, and adenine, just like DNA, and it also has uracil. Now remember, uracil in RNA acts like thymine in DNA. Thus, the uracil in RNA can link to the adenine in DNA. How does this help RNA take a snapshot of DNA? Think about it. The RNA can link its nucleotide bases to the bases on the DNA. The uracil in RNA can link to the adenine in DNA. The adenine in RNA can link to the thymine in DNA. The cytosine in RNA can link to the guanine in DNA. And the guanine in RNA can link to the cytosine in DNA. What does all this accomplish? When RNA does its job, it produces a negative of the DNA section that it is copying. Everywhere the DNA has a cytosine, the RNA will have a guanine. Everywhere the DNA has a guanine, the RNA will have a cytosine. Everywhere that DNA has a thymine, the RNA will have an adenine. And everywhere where DNA has an adenine, the RNA will have a uracil. This is much like what happens when film is developed. During the development process, the first thing to appear is a negative. This negative is dark everywhere that the picture is supposed to be light, and it is light everywhere that the picture is supposed to be dark. Based on this negative, then, a picture can be made. Based on the negative produced by RNA, a protein can be made. Before we show you how a protein can be made from this negative, we want to make sure you understand what we have discussed so far by showing you an illustration of it. When the RNA strand is built, then, it is a negative image of the DNA strand to which it is linked. This process is called transcription, and the negative image produced by transcription can be used by the ribosome to make a protein. Before we show you how that is done, however, make sure you understand the process of transcription by answering the following questions. Protein synthesis part 2. Translation. After transcription, the RNA leaves the nucleus and moves to the ribosome. This carries the negative image of the gene that was transcribed to the organelle that produces the protein. Because the RNA that performs transcription is essentially a messenger, sending instructions from the nuclear DNA to the ribosome, we usually call it messenger RNA, mRNA. Messenger RNA, the RNA that performs transcription. Now, if you're getting a little bit confused at this point, don't worry. We'll sum it all up in a nice figure at the end. For right now, though, just understand that in order to get the DNA's instructions from the nucleus to the ribosome, RNA produces a negative image of the DNA's nucleotide sequence and takes it to the ribosome. The ribosome is surrounded by amino acids, enzymes, and a different kind of RNA called transfer RNA, tRNA. Transfer RNA is a big molecule that contains a special sequence of 3 nucleotides called an anticodon. Anticodon, a three-nucleotide base sequence on tRNA. In addition to this anticodon, tRNA, or transfer RNA, is bonded to an amino acid. The amino acid bonded to the transfer RNA is determined by the three nucleotide bases in the anticodon. For example, when the anticodon is made up of uracil, and cytosine in that order, the tRNA is bonded to the amino acid lysine. If instead the anticodon is made up of uracil, cytosine, and cytosine, the transfer RNA is bonded to the amino acid arginine. This bond means that everywhere transfer RNA goes, the amino acid goes as well. When an mRNA molecule comes to the ribosome, the tRNA strands are attracted to the three nucleotide bases that will bond with the three nucleotide bases in their anticodon. Thus, if a tRNA strand has guanine, cytosine, and adenine, it will be attracted to a section of the mRNA that has cytosine, guanine, and uracil in succession. If such a sequence exists, the tRNA will go and link up with it, carrying the amino acid to which it is bonded the whole way. The three nucleotide base sequence that attracts the tRNA anticodon is called, reasonably enough, a codon. Since a codon attracts a tRNA with a particular amino acid on it, we say that a codon refers to a specific amino acid. Codon, a sequence of three nucleotide bases on mRNA that refers to a specific amino acid. Now, don't get lost in all of this terminology. A strand of mRNA can be thought of as a bunch of three nucleotide sequences. Each three nucleotide base sequence is called a codon. A strand of tRNA contains a three nucleotide base sequence called an anticodon. A certain anticodon on tRNA results in a certain amino acid bonded to the tRNA. Since the tRNA anticodons are attracted by the mRNA codons, the net result is that a codon on mRNA attracts a specific amino acid. The figure below illustrates this process. Now, look at what happens. When the tRNA strands have linked up to the mRNA, there are amino acids lined right up next to each other. What, by the way, is a protein? It is a bunch of amino acids linked together in a particular sequence. Well, at this point, the amino acids sitting right next to each other link up. This happens again and again and again, so that many amino acids link up together. When all the amino acids called for by the codons on mRNA are linked together, the result is a protein. The entire process is shown in figure 7.2. It's called translation. Now that we've been through the process once, let's summarize it so that you can review what you have learned. When a cell needs to make a protein, Its DNA has the plan for making that protein in a long series of 3 nucleotide base sequences. Messenger RNA reads the sequence and makes its negative image of the relevant portion of DNA. It then takes the series of nucleotide base sequences out to the ribosome. Once at the ribosome, each codon, set of 3 nucleotide bases, on the mRNA will attract a particular anticodon, set of 3 nucleotide bases, on tRNA. The tRNA that is attracted to the codon has a particular amino acid riding on it. This amino acid is determined by the anticodon on the tRNA. Thus, since each codon on mRNA attracts a particular anticodon, the codons in effect attract a particular amino acid. As the amino acids line up, they link together, eventually forming the protein. The process we just reviewed is split into 2 phases: transcription and translation. In transcription, mRNA makes its negative image in order to copy the nucleotide sequence in DNA. This is much like a transcriptionist copying a conversation by writing everything down. In the next step, translation, the mRNA leaves the nucleus and goes to the ribosome, where tRNA strands carry amino acids to the mRNA and line them up in the order determined by the sequence of nucleotides. The amino acids then bond together, making a protein. This step is called translation because the language of nucleotide-based sequences in RNA is translated into the language of amino acid sequences in a protein. Thus, as a bare bones description of how a cell makes a protein, we could say DNA through transcription leads to RNA through translation yields a protein. Whew. Now, of course, this doesn't show you the details of transcription and translation. Those are summarized in the figure below. Now, before we end this section, we want you to be aware that we have simplified this process a great deal in order to make it easier for you to learn. There are a lot of things that make this process more complicated, but we did not want to interfere with your getting a basic understanding of how a gene is translated into a protein. However, we do want to illustrate the level of complexity by just mentioning 2 things in brief. First, a given amino acid can be called for by several different codons. For example, the amino acid cytosine or cysteine can be called for by a sequence of uracil, guanine, and cytosine, or by a sequence of uracil, guanine, and uracil. Of course, a single codon cannot call for more than 1 amino acid, but a single amino acid can have many codons that call for it. In addition, there are certain things that have to happen between transcription and translation. For example, in eukaryotic cells, the mRNA that comes from the nucleus actually has too much information on it. You see, a gene in a eukaryotic cell contains sections called exons and other sections called introns. When the mRNA makes its negative image, it copies everything on the gene. However, only the exons are a part of the instructions for making a protein. The introns are sequences of nucleotide bases that separate the exons, almost like spacers that separate one set of books from another set of books on a shelf. In order for the ribosome to have the proper instructions, the mRNA must be processed before it gets to the ribosome. During this processing, the introns are removed and the exons are spliced together so that by the time it gets to the ribosome, the mRNA has the information necessary to make the protein, the exons, and nothing more. No introns. If the introns are removed from the mRNA before the protein is made, what is their purpose? The short answer is that we are not sure. Since they are not a part of the instructions for the protein that the gene produces, some scientists call them junk DNA. However, we know that God doesn't make junk, so introns must have a function. Indeed, current research is pointing to that fact already. For example, research indicates that different cells can use the same gene to produce different proteins because they can keep certain exons from the gene and discard other exons. Thus, because the introns split the protein making instructions into pieces, one gene can actually code for 2 or more proteins based on what pieces are used by the cell. This makes the genetic information even more compact. Isn't God's creation incredible? Now that you know what a gene is and how it codes for the production of a protein, your next question should be, how are genes passed on from one generation to another? After all, we know that offspring receive traits from their parents, and we know that traits are governed, to one extent or another, by genes. Thus, the genes of the parents must be transferred to the genes of the offspring. How does that happen? Well, to understand how that happens, you need to understand more about cellular reproduction. As a result, the rest of this module will be devoted to how eukaryotic cells asexually reproduce, as well as the way in which eukaryotic cells are made for the purpose of sexual reproduction. Once you learn these details, you will finally be able to understand how genes are passed on from one generation to the next. And that will be the focus of module number 8. Okay, friends, will you please like, and maybe even subscribe to my channel so that more students can find us? Oh, I really appreciate it. Thanks so much. We'll see you back in just a little bit with part 2.